Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Mast cell, it is a bone marrow derived large mononuclear tissue resident innate immune cell with special secretory properties. Coming to its embryology, it is derived from bone marrow, from hematopoietic stem cell which gives rise to myeloid stem cell which in turn gives rise to mast cell precursor. These precursor cells are immature cells without any granules. They will enter the bloodstream from bone marrow. Soon they will reach finally tissues where they become a mature cell with granules under the influence of stem cell factor interleukin 3 within those tissues where they will become a mature final mast cell. So even though mast cell is derived from bone marrow, it won't like to roam in the blood along with other blood cells. It will reach tissues and will settle there. Where they will settle and what they secrete, based on that, it is divided into two types. Type 1, they will settle in connective tissue. They will secrete kinase and tryptase, both the enzymes. Type 2 mast cell secretes only tryptase, settles down in mucosa of GI tract, respiratory tract, conjunctiva. Whereas type 1 mast cell settles in connective tissues like submucosa of GI tract, respiratory tract and dermis. Abundantly in the papillary dermis around blood vessels, nerves and appendages. Coming to its demonstration, on routine HNE stain, as it cannot stain the granules, this cytoplasm will look pale and empty with a dark central round nucleus, giving rise to a fried egg appearance. Most specific immunohistochemical marker of mast cell is tryptase. And other marker is c-KIT, also known as CD117. Coming to special stain, as HND does not stain granules, our special stains can stain granules where they show metachromasia and may obscure the nucleus. What do you mean by metachromasia? We will use one color stain, but the resultant stain will be some other color. Violet or reddish purple stained granules will be visible to us. Coming to electron microscopy, this cell is a round to spindle cell with large long villi at periphery and a central round to oval nucleus. Here you can see round to spindle cell with large long peripheral villi with central round to oval nucleus, with its cytoplasm containing numerous membrane bound electron dense granules. Coming to triggers of mast cell. Triggers are divided into two types. One is immunological. Main thing under immunological list is IgE antibody with its receptor Fc epsilon R1 on mast cells. Whenever a true allergen enters the body, it will bind to IgE antibody, which in turn cross-links Fc epsilon R1 receptor, leading to activation of mast cell. Under autoimmune chronic spontaneous urticaria, our body will produce antibodies against mainly IgE antibody and Fc epsilon R1 receptor, leading to stimulation of mast cell without any external trigger. That's why it is called spontaneous urticaria. Next, C3A, C5A complements will activate the mast cell using C3A, C5A receptors on the mast cell. Any infections, any autoimmune condition will stimulate the immune system to form immune complexes. Those immune complexes may stimulate these complements, or certain venom may stimulate directly through alternative pathway. Coming to non-immunological triggers, pseudo-allergens. True allergens require IgE antibody. True allergens will stimulate our immune system, whereas pseudo-allergen does not depend on our immunity or IgE antibody. They will have their own receptors on mast cell. They will directly activate the mast cell. Physical stimuli, cytokines, chemokines, neuropeptides, hormones, infections, infestations and venoms will directly activate mast cell via toll-like receptor. Stem cell factor will act via KIT receptor. to convert an immature mast cell to mature cell during its development. Upon activation by all these various triggers, mast cells will secrete several mediators via two types. One is degranulation. Already within those granules there will be several preformed mediators which will be immediately released from granules. For example, histamine, serotonin, heparin, hyaluronic acid, kinase, tryptase, VIP, substance P. All these are released immediately because they are preformed and stored in granules. Another thing is de novo synthesis. Mast cells upon activation they will start synthesizing freshly, so there will be delay in its release. For example, prostaglandin D2, leukotrienes like B4, C4, D4, cytokines, chemokines, growth factors. All these are because of de novo synthesis. Coming to applied aspects, mastocytosis because of KIT gene defect. It is of two types. One is systemic. If those abnormal mast cells gets accumulated in any of the internal organs, if they gets accumulated on the skin, it is called cutaneous mastocytosis, also called as urticaria pigmentosa. Mast cell activation syndrome, anaphylaxis, angioedema, allergic rhinitis, allergic asthma, and finally urticaria. Remember the channel name. Don't forget to like, share and subscribe. Thank you. Hasta la vista. До свидания.